Yes. Yes, we are going to share the presentation. Because we have a double audience. An audience here. Okay. Now we are ready.
So just to give you an introduction of what we are going to cover today. I use this slide just as a main reference for discussion. So whenever either during the discussion or after, we can use those as a reference if you prefer. The whole idea of NIGIDIA, which stands for Next Generation Diagnostics, was to put together some key feature that we found over the years harboring in our local ecosystem. So we have been a partnership, in fact, between the Teleton Foundation, the University of Naples, and some private funders. The Teleton Foundation that I don't know if many of you are aware of is the major Italian charity devoted to the study of genetics and rare genetic diseases. Essentially, over the 40 years of activity, the principal milestones are on the left. So we gathered almost €500 million in donations, thousands of papers, but also we got orphan drug designation and EMEA market distribution for gene therapy drugs. But behind the activity of NIGIDIA, there are 90% of professors from this university. So it's a partnership that allowed the flexibility of a private entity with the scientific concentration that only a university can put together and maintain. So NIGIDIA was then born to apply those milestones in research to life sciences, supporting the academic activity, perform clinical studies, particularly using next generation sequencing, so the approaches devoted to the study of our genome to understand and diagnose diseases, and finally, identify novel biomarker. Why? Because in the biotechnological field, but especially in the diagnostic field, there's a huge gap between when an application is used in research and when this is applied to the clinics. It is true for any innovation in the medical field, but it's particularly true whenever we have to deal with data. And the other barriers that are on the top is that everything is extremely expensive, so it doesn't fit with the welfare healthcare systems. It requires expertise that is difficult to put at scale. So the use of artificial intelligence in what I'm going to show you allowed us to multiply something that otherwise would require physical expertise of professional individuals. And finally, it's difficult to some of the results to become agnostic, let's put this way, to be translated from one field to another. Because whenever we talk about clinical practice, we talk about appropriateness. So a drug or any diagnostic device is specifically developed for an application. To use that tool, device, or drug, how do we call it, off-label, requires pretty much to start over the process if you want to be part of the health system. So the mission of NIGIDIA is to enable precision medicines, moving from research towards diagnostics. And one of the major applications that we had over the years to prove that implementing technology can tackle disease was during COVID. In COVID, the regulatory grid got a little bit lifted or loosened, so allowed us to pretty much go a little bit around the regulation just because of the urgency. In this case, for example, we developed a technology that allows us to sequence the whole COVID genome from the actual cost of €150 down to €20 per genome. So the whole sequence to identify the so-called famous variants. So this allowed Regione Campania to be the first Italian region sequencing half of the whole COVID genome running in Italy. So our institute, made of pretty much, and the activity composed of 3 scientists, was able to sequence more COVID genome than the entire hospital all over Italy. So this is an example of how technology, when it's, of course, put at its maximum capacity, can perform. But of course, that's not possible because we have regulations and we have to follow them. So in pathology, especially, whenever you want to get to a diagnosis, you get a biopsy. You have to provide a biopsy of the sample for analysis. This requires analysis with individual markers made by a pathologist, so somebody with a degree in medicine plus 5 years of specialization. So an extremely complicated analysis made by a highly professionally trained professional. So the idea is that we are trying in the field to go then from single gene tests that require that have analytical issues related to the expertise of the operator and at the same time of the human error that are still present whenever you do a medical activity, to something that we hope to be more comprehensive. So use tests and artificial intelligence not to replace the medical professionals, but to allow them to instruct easier diagnostics treatments. So increasing the quality and decreasing the cost at the end. So this comes from our experience that all over the years, we processed more than 15,000 samples, which is an enormous amount for a non-clinical centers and a small company like Nigeria. This because, as I said, in the normal pathology workflow, you have a first evaluation of you have to take your sample, make laboratory processing, cut the sample, do one marker at a time, so one gene at a time to analyze, review again the results and make the report. So it's a process that per se is expensive, time-consuming, difficult to scale, analogic, and provide limited knowledge in a way that you analyze one gene at a time. We have 20,000 genes in our genome, and in pathology, there are at least 200, 300 genes that are informative to disease. So with the RNA sequencing, so the sequencing of the RNA, which is the product of what our gene express in the cell, we can analyze in one assay all the gene expressed in the cell, pretty much 12,000. So our hope would be to go towards a digital diagnosis where you convert all the tests, all the need of the pathology lab to only one test to ease the work. And of course, we have proven that this overall has a lower cost, is fast, scalable, digital, and comprehensive. So this is a healthy circle, because then when you get the data, so in the AI view of the system, when you get the sample, you get the data feed a machine learning approach, you validate the result, you go again, the new sample, we create new data that will improve the model, and the model will improve itself over time. So you have understood that then, even for those of you that might have never heard of what genetic is, so the key needs for any AI application are data. In the case of genetics, are genetic data. So our genetic makeup that allows both to set baseline, so to set what in our genome is healthy and what was not. So here in the audience, we see people with different type of hair, length of hair, color of the skin, height, weight, and many other features. Those are the ones that we see. So those doesn't make us sick. It just makes us diverse. So in our genome, those elements are called polymorphic variants. So something that are variation of the genome that make us just different. It doesn't make us disease or not. So this is the baseline that we still have to fully understood to consider what is considered healthy and what is considered normal variation. Nevertheless, we need genetic data to do that. To give you an idea of how many data our genome contains in one cell, so you don't see your cell by eye, your cell is extremely tiny. In your cell, there are each nucleus of our cell contain about 3 billion bases. So 3 billion bases means that you take a book of 60 letters per line, 20 lines per page, and 2,000 pages, pretty much the Bible, all the New Testament. So you multiply this for about 1,000, and this is the amount of letters that are in one in the DNA of one of our cells. Our body is made of trillions of cells. Only 1-3% of this genome encodes for genes, so the element responsible to make up our body. The rest, it was thought to be useless, junk DNA. In reality, it has all the regulatory function to control that 1-3%. So this is the amount of, and then of course, you might know that the letters of the DNA is not a binary system, it's a quaternary system, A, G, C, T. So the alternation of this makes our genetic makeup. To give you an amount of, the amount of real data in terabytes, so how much our machine has to handle, consider that in one cell we have those 3 billion bases. That makes one whole genome. The sequencer of today can make, depending where you go to the US option or the Asian option, you can get in one day between 120 and 800 whole genome sequences per day, cover each genome at least 30 times per base. In amount of data, we're talking about 100 terabytes of data per day, which in reality is the entire Netflix catalog in terms of data produced every day by one of those sequencers. So you imagine that the amount of data, it's huge. In fact, now the new issue, in fact, you might have heard that now the money is not anymore in the samples, it's in the data. And the issue is becoming now how to store this data, because the data needs to be also always preserved in their rawest version. So as raw as possible to be always reanalyzed. So coming back to our huge mountain of genetic data that we need to provide, and I hope you got the sense of how much data we need to dig into. So we know very, very little of how our genome works. And in fact, 60-70% of the variant that we found in the population we cannot classify as passenger or disease causing, and this is a major issue in medical genetics. But depending on the regulatory system that we live in, we have other issues that prevent, that limits, but most likely prevent the use of genetic data to train AI systems. First one, and I think we can also stop and start discussing here. So if you want, if we have time, we can show what Nigeria did, how we train the systems, but I think it's more useful to discuss today about this point. Data privacy law. In Europe, we have the stringest data privacy law in the world. So we cannot take people information if we don't have the consent given in a specific way. The data need to be interoperable, accessible, because anytime you can say, take out my data of your analysis. Unfortunately, when we talk about health data, we need approval from ethical board. So while in the US you might have heard about 23andMe, MyHeritage, customer provide, and it's called business to customer approach to genomics. So where you can give your DNA and they identify your heritage, your ancestry, your predisposition to disease. It's very nice. I've done it 15 years ago. Unfortunately, according to the existing European law, you cannot do it. To be able to take somebody's DNA, their genetic information, you need to have, besides the consent, the informed consent, what you're going to see, what you're going to find, your risk that you might see. 10% when you do a genetic test for the genetic disease, 10% of the fathers are not the fathers. So this information can change somebody's life. And that's the reason why we need to have extra protection. The local ethical board, so you can touch somebody's DNA only for diagnostics, so the clinical question needs to be provided and clear, or for research. For research, you need to have local ethical board approval, which is a nightmare, because of course they are the gatekeeper to be sure that you can use the data for a specific purpose, for a specific time, for a specific approach. You cannot use for any other way. You have to. So it's a process. So you cannot just take, but this is for DNA, but also for the medical record. You cannot just take all the medical record of an hospital and just feed a machine learning tool. We can debate whether this is right, this is wrong. There are systems where they don't care about that. And in fact, they are evolving much faster. In order, China, UK, US. UK has an extremely light approach to privacy law. What UK is doing, for example, the UK Biobank, is today inapplicable to the European Union. Third, and connected to the others, data residency. So all the providers all along the way need to are responsible that the data resides in a country, in the case of Europe, in a European countries. Depending on the data, it needs to reside also in the country itself. So not an EU country count as the same. For genetic data is even more important. So generally you give your consent to an owner of the consent, what we call first owner of the consent. And this person is able to nominate second or third responsible of the data. For genetic data, everybody is the primary owner of the consent. In this way, you can be pursued by the law directly without saying, oh, I didn't know if somebody give me the consent in this way. It's because our legal system put so much responsibility because the genetic data is somebody that allow me to identify you anywhere in the world. If I have 25 markers of your DNA, and I can identify you, your generation up to 8 generation below or above. So with an error rate of 1 out of 6 billion, so more of the human population. And finally, the certification. So likely or unlikely for us, the European Union transition now to a new certification for in vitro medical devices, which requires also that you cannot just use an AI tool to analyze the data. To analyze the data in a way that's acceptable for diagnosis needs to be under this CIVDR new regulation of the European Union. So it needs to become a certified tool that can be run only on certified centers that follow the requirement above. And the lab itself needs to have extra certification. Some countries, as I said, are in the US, you can self-register a medical device. So you know that those FDA tool, it was possible still in Europe until 4 or 5 years ago, maybe less. Now it's not possible anymore. So, and that's the reason why it's easy. So sometimes drugs, but medical devices gets out of the market easier in the American system. And the same drug takes then 10 years more